Thank you very much for having me at this fabulous conference. My name is Liachko. I run Phage-Genomics. It's a company in Seattle, Washington in the US. And so what I'm going to talk about today is a technology we've developed that really solves one of the key difficulties in the study of microbiome and therefore the holobiome. And that is the simple fact that if you're studying any kind of microbial community, any kind of mixed community, maybe it's not even microbial, that you're dealing with an issue where the moment you purify DNA to sequence it, it's mixed, right? And so we've heard a lot about this already, like Professor Sorensen discussed today, right? If you have a plasmid in one of your microbes, the moment you've purified DNA, you can never get that host information back directly, right? If you have a phage or a plasmid, once you have a shotgun metagenome, you don't know where it came from anymore. And of course, that compounds the problem on top of, having to deal with multiple genomes that you're trying to reconstruct at the same time. So what we've done is we've repurposed a method that was originally designed for something else. It was originally designed to look at how a genome folds in three dimensions, and we use it to basically solve this metagenomic problem. So the idea is if you can take microbial community and treat it with formalin. Formalin or formaldehyde, right, is a cross-linking agent. It goes into the cells and it creates physical junctions between DNA sequences that are close together. So it cross-links proteins, RNA, DNA, and it immobilizes everything in its sort of three-dimensional structure. And in doing so, essentially, the simplified cartoon, it traps sequences that are physically close together in three-dimensional space, which you can then, through a series of molecular steps, you can join them, creating these chimeric sequences between molecules that are next to each other, and then you can turn them into Illumina libraries and sequence them. So essentially what you're doing is you're doing Illumina paired-end sequencing on not a linear piece of DNA, but two pieces that are linked together. And you know that whatever the sequence is, this red sequence is, and whatever this blue sequence is, they must have been physically close together in 3 dimensions. But remember, the cross-linking happened before you broke the cells. So if two sequences are forming this junction, they must have been inside the same cell. Okay? So any two sequences that interact by this assay must have been inside the same cell at the beginning of the experiment. And so what you can do now is now you have your metagenome and you know, right, which pieces are touching each other. You're essentially saying, I know DNA sequences that are touching each other, and you get both intra-chromosomal and inter-chromosomal junctions, okay? And so you're able to then take your metagenome and essentially separate apart genomes based on not sequence or abundance, but actually you know that these sequences were physically touching each other. And so, and on top of that, you will not only get junctions within chromosomes, you're also going to get plasmids and phages interacting physically with their hosts. So effectively, it's a short-read data set that does this, right? That it connects pieces of genomes together, and it also connects mobile elements directly to their hosts without special machinery or anything like that, just good old short-read sequencing. So it does a few things. So it's a way of binning metagenomes in the way that you just can't do with regular binning. So here's a paper we published with folks at the USDA and PacBio, and I think some folks in this audience. This is a single sample that we've sequenced. This is a sheep fecal sample. You get 400 mags genomes that are over 90% complete, 200 lineage resolved genomes, but also 400 phage-host interactions and 300 plasmid-host interactions, right? All from the same sample. So you're basically getting genome reconstructions of the hosts as well as of the plasmids. In fact, we've done a lot of other work that aren't entirely microbiome. So we've done a lot of these kind of hologenome projects where you're simultaneously sequencing by this assay, like an arthropod and its microbiome, and you can separate them out. So I saw a poster outside that folks are doing using this proximity ligation technology to assemble genomes. Remember, you can use it not only to assemble genomes, but to separate the microbial community from the host as well. So, but this just shows you how much of this kind of information you can get from even single samples. This is data from collaboration with Dr. Lindsay Edwards, who's in the audience. This is basically looking at just a brief snapshot of a healthy gut sample from an FMT experiment. So all the little circles are bacteria or microbes, right? So all the little stars are microbes, and then orange Orange diamonds are plasmids, and blue triangles are phages. So you're simultaneously de novo assembling phages, assembling plasmids, and connecting them all together. And I show this because this is how we're used to looking at microbial communities, at metagenomic data. But really, your metagenome looks like this, right? That's what's actually happening inside of these communities. And we don't see that by normal sequencing methods. And so that sort of comes into play when we're talking about the holobiome, because it's essentially the same thing, right? This is sort of like one level down. Just like when people sequence the human genome, they don't think about the microbiome. Well, when you sequence the microbiome, do you think about the mobile elements? Because that's another level as part of this, right? Now, we don't only work on humans. So Ben, my colleague, Ben Auk, is going to talk tomorrow about this kind of approach to looking at fecal transplants and sort of clinical applications of this technology, what happens in these microbial communities to the mobile elements. But we also do a lot of work in other environments. So for example, this is a rumen sample. So we're doing a lot of work on cattle rumen and methanogens. And I show it mostly to highlight, like, look at how many there are, right? This is a single sample. There are thousands, right? There's over 1,000 phage-host interactions in here. All the little blue things are phages. And so we're getting tons and tons of phage information, lots of plasmids, and they're almost all novel because, again, unless you're sequencing the human gut, most microbiota is unstudied. And so we now have, we've gotten some funding from the Gates Foundation and NIH. We've built what is the world's largest collection of phage-host interaction data because we've been processing samples, thousands of samples from fecal, from water, soil, et cetera. So we have something like 2 million phages in our database. And when you have so many phages, what are you going to do with them? They're sort of all over the microbiome. They're not cultured, right? So you're getting all sorts of crazy organisms that nobody normally would pay attention to. So what I'll do is I'll show a couple of quick slides about something interesting that we're doing. So when people think about phages, obviously a lot of times they think about using them to fight antibiotic resistance, right, in phage therapy. That's like one of the cool applications du jour. But you forget, like we see these pictures in textbooks of phages just like bursting out of cells, but they don't just burst out of cells. They actually produce a protein called a lysine, and a lysine is what lyses the cell, okay? And these lysine proteins can be used as effective antibiotics. Here's a video from Vince Fischetti. What you're going to see is these are bacterial cells, and they're about to be exposed with some purified lysine protein. And just follow the arrow. Boop, gone. There's another one. Boop, there's one, pops. Right? There's no phage here. This is just a purified protein. And in about 10 seconds, it clears this field of bacteria, right? And so essentially a lysine is a species-specific antibiotic that overcomes known resistance mechanisms, and also it's very difficult for bacteria to develop resistance against lysins, unlike phages, because there's no phage replication cycle, so CRISPR will not help you, et cetera. And the other advantage of them is just the gene, right? So I told you we have 2 million phages in our database with hosts. That's the important thing. We actually know who they're infecting, right? And so now we're doing things ourselves where we're sort of isolating lysins and testing them, and it's pretty cool. Like a lot of them are just purifying them out of our database, just predicting them out of our database. They can become quite powerful. I wanted to finish this talk with this. I just saw this article like 2 days ago. Apparently, Denmark's about to start putting extra taxes on cow burps. We just got some funding from Gates Foundation and the Grantham Foundation to try to use lysins to knock down methanogens. And so this is some pilot data in collaboration with a company called Lumen Bio. Essentially, you can see there's a few candidates that we pull out of our database that knock down methane by like 95%. Okay? And it's a very inexpensive way of doing it because, again, all you need is get a gene synthesized, express it, and go. So with that said, we're building a big antibiotics for everything kind of like foundry. Like we now have all these lysins. We're looking for targets to hit them with, and so to hit with them. So if you guys have interesting microbes, or even using them to modulate microbial communities, subtract certain components of a holobiome, things like that. And so with that, I will just move forward. This is a summary of the tech, and we sell it. This is a piece of software that we've just released called Explorer. If you're interested, let me know. And that's it. This is just a picture of us looking cute. Thank you.
Thank you very much. Thank you very much, Ivan.